Good morning, everyone. Welcome back. Today, I will talk about Western blotting technique. So, in my previous video, I already explained about the blotting technique and Southern blotting and Northern blotting technique. So, today, I will explain about Western blotting technique. So, in this video, I will be covering introduction, techniques, procedure, application, and disadvantages of Western blotting technique. So, introduction. Blots are techniques for transferring DNA, RNA, and proteins into a carrier so they can be separated and often follow the use of a gel electrophoresis. The southern blot is used for transferring DNA, the northern blot is used for RNA, and the western blot is used for the proteins. So types of blotting techniques are three: southern blot, northern blot, and western blot. Southern blot is used to detect DNA, northern blot is used to detect RNA, and western blot is used to detect proteins. Now talking about western blot, a technique for detecting specific proteins separated by electrophoresis by use of a labeled antibodies. Procedure: tissue preparation, cell electrophoresis, transfer, blocking, detection, and analysis. Now, talking about the first step, that is preparation of the tissue. Samples may be taken from whole tissue or from cell culture. In most cases, solid tissues are first broken down mechanically using a blender. It should be noted that bacteria, virus, or environmental samples can be a source of protein, and thus Western blotting is not restricted to cellular studies. Assorted detergents, salts, and buffers may be employed to encourage lysis of cells and to solubilize proteins. Tissue preparation is often done at cold temperature to avoid protein denaturation. Next step is gel electrophoresis. The proteins of the sample are separated using gel electrophoresis. Separation of protein may be by isoelectric point, molecular weight, electric charge, or a combination of these factors. Now you can see here in the diagram, it is shown how this technique performs. See how we are adding mixture of DNA molecule of different sizes in the gel, and which is connected to a power source, one end to the cathode and one end to the anode. So how it will separate as long molecules and shorter molecules. Next step is transferring. In order to make the proteins accessible to antibody detection, they are moved from within the gel into a membrane made up of nitrocellulose or PVDF, that is polyvinylidene difluoride. The membrane is placed on top of the gel and a stack of filter paper placed on top of it. The entire stack is placed in a buffer solution which moves up the paper by capillary action, bringing the proteins with it. Another method for transferring the protein is called electroblotting and uses an electric current to pull proteins from the gel into the PVDF or nitrocellulose membrane. Next step is blocking. The membrane has the ability to bind to the proteins. In this case, both the target and antibodies are proteins, and so there could be some unwanted bindings. Blocking of non-specific binding is achieved by placing the membrane in a dilute solution of protein, typically BSA, that is bovine serum albumin, with a minute percentage of detergent such as Win20. The protein in the dilute solution attaches to the membrane in all places where the target proteins have not attached. Thus, when the antibody is attached, there is no room on the membrane for it to attach other than on the binding site of a specific target protein. Now, the another step is detection. There are two steps for the detection of protein. First, primary antibody, and second one is secondary antibody. After blocking a dilute solution of primary antibody, the solution is composed of buffered saline solution with a small percentage of detergent and sometimes with powdered milk. The antibody solution and the membrane can be sealed and incubated together for anywhere from 30 minutes to overnight. Now talking about secondary antibody, after rinsing the membrane to remove unbound primary antibody, the membrane is exposed to another antibody directed at a species specific portion of the primary antibody. Several secondary antibodies will bind to primary antibody and it will enhance the signal. See, as shown in this figure, membrane with transferred proteins, then we are adding blocking buffer, then primary antibodies added, incubate blot overnight at 4 degrees Celsius. After that, wash buffer, then the addition of TBST. After that, horseradish peroxide conjugated secondary metabolites are added. Incubate blot for 2 hours at room temperature. Again, TBST is added and membrane is ready to visualize. So this is the whole process. Now how we can perform the analysis? Different techniques are used, like calorimetry detection. The calorimetry detection method depends on incubation of the western blot with a substrate that react with a reporter enzyme such as peroxidase. Another technique is chemiluminescent technique. This method depends on incubation of the western blot with a substrate that will luminescent when exposed to the reporter on the second antibody. Another technique is radioactive detection. Radioactive labels do not require enzyme substrate but rather allow the placement of the medical X-ray film directly against the western blot, which develops as it is exposed to the label and create dark region which correspond to the protein band of interest. Now talking about the applications of western blot, the confirmatory HIV employs a western blot to detect anti-HIV antibody in human serum sample. Proteins from known HIV infected cells are separated and blotted on a membrane. Then the serum to be tested is applied to the primary metabolite. Primary antibody incubated step. Free antibody is washed away and the secondary anti-human antibody linked to an enzyme signal is added. The stain bands then indicate the protein to which the patient serum contains the antibody. Western blot can also be used as a confirmatory test for hepatitis B infection. Now talking about the disadvantages of western blotting. If a protein is degraded quickly, western blotting would not detect it well. This test takes longer than other existing tests. It might also be more costlier as compared to the another techniques. So this was all about western blotting technique. Hope you understood about western blotting technique. Thank you.